Hello, my name is Danielle Snowflack, and I am the Director of Education, Professional Development, and Outreach at the American Society for Biochemistry and Molecular Biology, or ASBMB. The ASBMB is a nonprofit scientific and educational organization with over 12,000 members worldwide. Founded in 1906, the society is based in Rockville, Maryland, and publishes the Journal of Biological Chemistry, Molecular and Cellular Proteomics, and the Journal of Lipid Research. I'd like to welcome you all to today's webinar, Small Business Research Funding 101 with the NCI SBIR Development Center, which is hosted by the ASBMB Education and Professional Development Committee. This webinar focuses on funding opportunities and other commercialization resources for small businesses in biochemistry and molecular biology. If you are having problems connecting to the webinar audio through your computer, you can connect via phone at the number on the slide that you should see right now. If you connect via phone, you must mute your phone throughout the entirety of the webinar. All phone users can be heard within the webinar suite, and we will have extreme difficulty delivering the webinar to all of the participants if we have background noise. Before we get started, I'd like to direct you to a few features in the webinar room. First, there is a poll at the top right of the screen. This poll will give us a little information about your stage in applying for SBIR funding, which will help us cover topics that are most important to you in our discussions. Second, throughout the presentation today, we will be accepting questions and sharing resources. You can submit a question through the Q&A box at the bottom right of your screen. In the interest of time, we will hold questions until the conclusion of the presentation. At this time, the speakers will answer previously submitted questions, as well as new questions that may arise. With that, let's get started. I'd like to introduce you all to Dr. Jenna Hendershot, who is a senior scientist in new product development at Kaman Chemical. She's also a member of the ASBMB Education and Professional Development Committee and the chair of the EPD Graduate and Postdoc Subcommittee. Jenna, would you like to provide a welcome on behalf of the ASBMB?
Thank you very much, Danielle. As you just heard, my name is Jenna Hendershot, and I want to thank everyone for attending our webinar today. So the main goal of the Education and Professional Development Committee, and specifically the Graduate Student and Postdoc Subcommittee, is to make career and professional development resources. And so we've been really happy with the success of our webinar series, and I just wanted to let you know that you can check out all of our previous webinars on the ASBMB YouTube page at any time. So I'm very excited for our webinar today. We have two great speakers, and on behalf of the ASBMB, thank you very much for joining us.
Thank you so much, Jenna. Next, I would like to introduce you all to Dr. Kory Hallett, Program Director in the SBIR Sttr office. Kory?
Thank you. I'm Kory Hallett. I'm a Program Director here at the NCI in the SBIR and Sttr office. I've been here for about 5 years. My portfolio is mostly in the area of immunotherapeutics, but I can often answer questions. I can answer questions about the program generally. I can sometimes answer questions about other technology areas. And I will be speaking with you more about our program in just a few minutes.
Great. Thank you so much, Kory. Next, I would like to introduce you all to Dr. Sami Kanaan, President and co-founder at Chimerocyte in Seattle, Washington, and staff scientist at the Fred Hutchinson Cancer Research Center in Seattle, Washington. Sami?
Hi, I'm a scientist. I did my PhD in France, and I came here 5 years ago to work with Dr. Lee Nelson. And last year, we decided to, we're working on a technology, and we decided to spin it out into a clinical application. And this is how we applied for an SBIR Sttr grant, and I'll talk about that more. So yes, welcome everyone.
Great. Well, thank you so much, both of you, for joining us. And with that, I am going to move to the slideshow. Kory.
Yeah, thank you. I saw one or two people say they were having a hard time hearing me in the chat, so I've turned up my microphone volume, and hopefully this is easier. Please let me know. I can see the chat, so please let me know if you can't hear. I'm Kory Hallett, a Program Director in the NCI SBIR office. I'm going to give you, in this presentation, just a very high-level overview of what the SBIR and Sttr programs are. I'll try to give you a few specifics, but if there's one take-home message from this today, it's if you're interested in small business funding and you're thinking about the SBIR and STTR program, don't hesitate to reach out to us. My email is right there on the main slide. You can email me directly. On the thank you slide, I think will be our general inbox, and we can always connect you with the right program director. If I'm not the right program director, I'll connect you. If you email the general inbox, someone else will connect you. So the SBIR and STTR programs are congressionally mandated programs. What this means is that there is a set aside, so a certain percentage, it's currently 3.2% of the NIH budget is set aside for the SBIR program, and 0.45% of the NIH budget is set aside for the STTR program. These two programs, I refer to them as sister programs. They're administered together. Many of the rules are the same between the two programs, not completely, but many of them. The SBIR program is specific for small businesses to engage in R&D with a focus on commercialization. The STTR program also is specific for small businesses to engage in R&D, but small businesses that have a close relationship with a not-for-profit research institution, often a university, and they're pulling that technology out of the university and moving it forward for commercialization. The NIH is not the only agency with an SBIR and STTR program. You can see there are several agencies listed on this slide that also have SBIR programs. Our budget at the NIH, we're the second largest SBIR and STTR program across the federal government. DoD is the largest, and HHS, which is mainly NIH for this program, is the second largest at just over $1 billion per year given to small businesses for R&D. At NCI, $167 million for fiscal year 2019. We will be awarding to small businesses. I keep hitting my arrows on my keyboard. As I mentioned, the programs are sister programs. They're very similar. There are a few critical differences. The under the SBIR program, the PI that's leading the project must be an employee of the small business. So 50%, more than 50% of their employment status must be with the small business. They're allowed to partner with a university, but it is not mandatory. And therefore, they're allowed a small amount of their award dollars can go to a university, but not the whole thing. The STTR program, because that is a program specifically for small business that's working closely with a not-for-profit research institution, the PI does not need to be an employee of the small business. PI employment's not stipulated. They must dedicate 10% effort to the project, but if this is a faculty member or a scientist at a university spinning out a small business, this is the right program to apply to. Partnering is required between the small business and the not-for-profit research institution. And then the small business must do 40% of the work, and the not-for-profit must do 30% of the work, and then the additional 30% can go to either party. So under the STTR program, a university could do up to 60% of the work on a phase 1 SBIR. Under the STTR, because there has to be this agreement between the not-for-profit and the small business, there must be a written agreement stating that the small business has the rights to take the technology forward for commercialization. We generally recommend you do this in SBIR also if you have a close relationship with a university, but it's not required. The program is separated into 3 phases. These are not clinical trial phases. This is just how the legislation is written, that the grants come in phases. So a phase 1 is a proof of concept study. It's up to $300,000 for 6 months to a year. These are early stage in vitro assays. They can usually leading up to some, if it's a therapeutic, at least leading up to some early in vivo proof of efficacy. Phase 2 is $2 million over 2 years. This is continued research and development. For therapeutic projects, we often see people that are applying for phase 2 to complete some tox studies and some early IND enabling studies. There are some options depending on how mature the technology is. There's something called a fast track, which is a combined phase 1 and phase 2 application. There's also a direct to phase 2 option. I encourage everyone to reach out to a program director before they apply. especially if you're thinking of applying to a fast track or a direct phase 2. At NCI, we administer something called a phase 2 bridge application. It's a follow-on award to a phase 2. This is for $4 million over 3 years. This is to support it is to encourage small businesses to start seeking other sources of funding, because likely you will need more than the phase 1 and phase 2 amounts of money to take the product to commercialization. So one thing that is critical about the bridge is to review well under this program, you need to have substantial third-party investment funds to match the federal investment. There is a phase 3. This at NIH, a phase 3 is considered commercialization. It's the use of non-SBIR Sttr funds, but some other agencies like DoD do have a formal phase 3 as part of the program. So briefly, when are SBIR and Sttr grants appropriate? If you think about the timeline with an idea coming out of academia and some pre-seed funding and then seed funding and raising sort of money from friends and family, at phase 1, SBIR and Sttr is appropriate very early, sort of in that you're off the founder money, but you're moving on to friends and family and maybe some early angel investment. Phase 2 is sort of bridging angel investment over to some more institutional venture capital funds. And then the bridge award would be at the time when you're really seeking serious fundraising. This isn't a hard and fast rule. This is commonly the stages at which people are seeking SBIR funding. So why do people seek SBIR and Sttr funding? Well, the biggest reason is that it's not a loan and it's non-dilutive. So there's no repayment required. It is a grant like any other federal grant. And also the NIH does not own a piece of your company. And so it can be a really attractive way to get early seed stage money. Like other NIH grants, the intellectual property is retained by the small business. And some small businesses say that it helps with scientific verification and validation, and when they go on to seek investment funding from more institutional investors, the fact that they have received SBIR funding is a positive thing that they can talk about. Our applicants come from a variety of places. I have 2 examples on this slide. One, Lori Hazelhurst is a professor who was working on drug development with R01 funding, and as the drug matured to the point where she really wanted to take it forward for commercialization, she started a small business and spun it out of the university, and then started seeking SBIR funding to help her move that product forward. Aruna Gambier is another example of a successful small business owner that has received SBIR and Sttr funding, and she comes from a very industry heavy background, but was presented with an opportunity to start a small business with a product, and so she uses SBIR funding, understanding that she's going to seek private investment in the future. She uses the SBIR funding to de-risk her technology. Sami, who will be talking after me, he is also an SBIR funded small business. And so he can talk a little bit about his experience. So as I've sort of mentioned, but maybe not directly, the award is always made to the small business, and the small business must be eligible to receive SBIR funding. So the small business has to be for profit and has to be 500 or fewer employees. That's the federal definition of a small business. It also has to be owned and operated within the United States. This is a pretty strict criteria that the majority of company ownership must be by U.S. individuals. Our funding announcements differ a little bit from academic funding announcements. For grants, for SBIR and Sttr grants at NCI, most of our grants come in through what we term the omnibus grant solicitation. We don't specify specific technology areas that we're looking for under the omnibus. We want people to apply with all of their independent investigator-initiated ideas with their small business. There are 3 receipt deadlines per year: September 5th, January 5th, and April 5th. We do participate in some targeted funding opportunities. We have a handful of targeted grant funding announcements that you can find on our website. And then for NCI, most of our targeted funding announcements in the SBIR and SBTTR program come out as contract opportunities. Those come out in every fall, usually around late July, early August, and they're a one-time per year announcement. We fund a variety of technologies. A fair amount of our portfolio goes to therapeutics and other drugs, but we have several imaging technologies, in vitro diagnostics, non-regulated technologies like research tools, and then also we have a growing behavioral medicine and e-health portfolio. So I just show this slide to say that we are technology agnostic, and a lot of this is what comes in through the omnibus solicitation and peer reviews well. That's what dictates what percentage of our portfolio goes where. We have, there are several sort of small business development opportunities once you're a funded SBIR company. We have commercialization workshops. We have learning webinars. We call them the peer learning or plan webinars. I-Core at NIH is an entrepreneurial immersion course where small businesses that are phase 1 awardees, so they're still very early in their technology development path, they take a 10-week course where they're getting out and interviewing 100 potential stakeholders and trying to really understand where in the market their technology best fits so that they can use that information to take appropriate product development steps. We also have an investor initiatives program where small businesses that are funded by NCI SBIR can participate and have the opportunity for NCI to pay some showcase fees at different investor showcases throughout the country as they're trying to raise private capital. We put on targeted workshops as needed in different technology areas. And then NIH manages some market analysis and commercialization planning programs called the Niche Assessment Program and the Commercialization Assistance Program. All of these things are available to NCI SBIR awardees. We know that a lot of people are trying to get into the SBIR program, and the application in itself can be a bit of a burden, so we've started something called the Applicant Assistance Program. This program is free application preparation assistance. It's not a grant writer, but it is a specific coach that'll help you through registering your small business, understanding how to apply, and understanding the right way to draft your application. An SBIR and STTR application looks a lot like an R01 or an R21, but it needs to have a different focus because the focus really needs to be on the product. And so in this applicant assistance program, a coach will sort of read through it and give you feedback to make sure that you have that right sort of angle. The eligibility criteria are simple. It's just no previous NIH, SBIR, and STTR grants awarded. We have a particular interest in women-owned businesses, minority-owned businesses, and then businesses operating in underrepresented states, also known as IDEA states. We are running one cohort for the AAP in 2019. The application portal is currently open. I'll have it on the next slide. The application deadline is May 30th, and then the program will run from June to September, helping participants prepare their September 5th submission, or their application for the September 5th due date. So as I mentioned, the portal is currently open. The application is simple. There is a small series of structured questions. There are a few supporting documents, so an abstract and a statement of the current status of the technology, and then submit. I encourage everyone to look at this portal if they're interested in the AAP. Again, here's another link. We have a webinar specific for the AAP if people want to learn more on May 9th from 1 to 2, and you can find this by going on our website. Here's the program staff. As I mentioned in the beginning, if there's one thing to take away, it's don't hesitate to contact us. You can contact us and ask questions about the program. You can also contact us and send us your specific aims page, and we will look it over and give you feedback. Finally, I want to leave you with this, which is if you do seek SBIR funding, it's a competitive program. Always be prepared to resubmit. So after you apply to SBIR and you receive your summary statement and you receive your score, always contact your program officer and understand that the majority of people have to resubmit at least one time before they are successful. There's a lot of information on our website. Our website is sbir.cancer.gov. We are also pretty active on Twitter and have a LinkedIn page. So you can also sign up for our email lists. And I'll turn it back over to the host. Thank
you. Great. I will now turn it back over to Sami.
All right. Hello, everyone. So in this talk, I'll be talking a little bit science, so a little bit about the science of our technology, and then how we found a clinical unmet need, and we decided to go for the company, and then some word of how we got some assistance and what are we doing and where are we going with the company. All right, so let's begin. I'm going to be talking about application of chimerism testing or highly sensitive chimerism testing that we develop at our company. So I am Sami Kanaan. This is the disclosure. So I am a staff scientist at the Fred Hutchinson Cancer Research Center in Seattle, but I'm also president and co-founder at Chimerocyte, which develops solution for highly sensitive chimerism testing. Here's a little word about chimerism in normal human biology. So there's two types. You have the iatrogenic chimerism or treatment-related chimerism that comes from, for instance, transplantation, transfusion. Here's an example of a liver transplantation. And you also have the naturally acquired chimerism, which is usually done in small quantities. This is why we call it microchimerism, and it's the result of the bidirectional traffic of cells between the mother and the fetus. So the mom will have fetal microchimerism, and the child will have maternal microchimerism. And those cells can be found in all parts of the body, and they can have significant immunological effects. So it's very interesting to study them. How do we study chimerism? There's a few ways. For instance, there's a classical cytogenetics, including FISH or fluorescence in situ hybridization methods, where you can detect male in female or female in male cells. For instance, here, I don't know if it's visible, but you can see a female cell, which is XX, between X and Y. So the X is painted in pink and the Y is painted in green. And this is how we detect female microchimerism. We can do also PCR-based methods like the Y chromosome specific sequence, and we can detect male in female. If we want to detect any source of chimerism, there is, of course, the short tandem repeat or Sttr characterization PCR, which is currently the standard of care in hematopoietic cell transplantation. That is, used to monitor chimerism post-transplant. All those methods have, some problems. For instance, they have low sensitivities. For instance, this method is at best 1 to 5% sensitive. They have limited informativeness. For instance, here you can only detect male in female or female in male, but not male-male or female-female. Some of them are also laborious. So how do we do it in our lab? There's several steps. The first step is the most difficult one because we need to design highly sensitive and specific and quantitative PCR assays. Currently, over the years, we have developed several of them. They have a limit of detection of 1 in 100,000 to 1 in a million. Most of them have specificities to the MHC region or HLA in humans. And we also have other specificities outside the HLA region. For now, we have 6, but more are under development. And we have, in partnership with the Fred Hutchinson Cancer Research Center, put them in a patent. All right, so step 2, we can identify targets for chimerism. For instance, if we want to detect fetal microchimerism in a mother, for instance, we do HLA genotyping. So one will be shared between the mother and the fetus, but one will be not shared. So the green one here is the one inherited by the father. So here it's the DR7, and we just go and pick our assay from our panel, and then we run it. And then we analyze the data. And here is just to illustrate that we can be very sensitive, and we're confident about our sensitivity. For instance, here we can detect 6 cells in 1,000. Here we can detect 1 cell in 100,000. All right, so where are the areas of application of our highly sensitive chimerism test? So we can apply them in treatment-related chimerism. You have hematopoietic cell transplantation. You have also solid organ transplantation or transfusions. But also in the natural setting of microchimerism, we can apply them in several of our favorite fields in human biology and clinical research. So for instance, in our lab, so this is just work in our lab, we have described naturally acquired microchimerism in autoimmune diseases, including systemic sclerosis, rheumatoid arthritis, type 1 diabetes. In cancer, for instance, we demonstrated that fetal microchimerism is actually decreased in women that have breast cancer. So you can hypothesize that there's some sort of alloimmune surveillance that protects against breast cancer. In transplantation, so we studied maternal microchimerism in cord blood, and that cord blood will be used will be used in transplantation to cure leukemia, for instance, and there can be some benefit to that naturally present maternal microchimerism. In infectious disease, for instance, in collaboration with Whitney Harrington, we have described maternal microchimerism in the context of malaria, for instance, where those maternal cells can help reduce the symptoms of malaria. Also in pregnancy, in normal pregnancy and fetal maternal health, we have studied the effect of microchimerism on the normal course of pregnancy, but also complications, including miscarriage and preeclampsia. And in neurobiology, we're currently studying microchimerism, and we're detecting microchimerism in several parts of the brain. But I'm not going to talk about this right now. All right, in treatment-related chimerism, so we have done a collaboration here at the Fred Hutch, where we described chimerism that comes from double cord blood transplantation. So in adults, usually often, we need to transplant not only 1 cord blood, but 2, because cord blood sometimes do not have enough cells. And what happens often is that there's a mixed chimerism stage, and then 1 cord blood will become the sole source of hematopoiesis, and the other cord blood will vanish, and it's going to be called the losing cord blood. And we just demonstrated that the losing cord blood is not completely lost, but it's present at micro levels, and it could have some clinical significance. All right, so this is all chimerism in research, and what we wanted to do is to explore some clinical applications. And the first step was to find an unmet clinical need. So, for instance, chimerism is done routinely after transplantation. This is done all the time, but the method is old. It's since the 90s. The gold standard is the Sttr, as I talked earlier, which has a low sensitivity of 1 to 5%. And so there's a need to improve chimerism testing, and there's also a big need to improve methods of detecting minimal residual disease. So this is a problem in cancer, where even after transplantation, there's often a third of patients that we're gonna relapse. The idea is that chimerism is often correlated with relapse, in a way that the bone marrow should be 100% the donor source, and then any recipient cell that are coming back is a bad sign. So it's either a sign of the graft not engrafting correctly, or those minimal residual disease, so those leukemia cells are coming back. The idea is, the hypothesis was, what if we can test chimerism with such sensitivity that we can identify high-risk patients as early as possible and give that information for doctors to be able to treat better their patients. And so this is what we did. We applied for an Sttr grant in collaboration with Fred Hutchinson. This Sttr was awarded, and it could support the early phases of our site. And so it's me and my mentor here that we founded the company. And right now we're doing the very early phases. We're doing the proof of concept for our technology. All right. So what do we need to do? You know, how did we do it? How did it find help and support? So the first element here is support from the institution or university, and this, I think, is the critical part. We had huge help from our institution, the Fred Hutchinson, also the University of Washington, that has a dedicated program to help startup companies. Actually, our headquarters now are in the University of Washington, in a lab called Co-Motion Labs, where they host many startup companies coming from the consortium of the Fred Hutchinson and the University of Washington. We can also get some external help from nonprofit organizations. So for instance, in Seattle, there's many of them. One of them that was very beneficial to Chimerocyte was Life Science Washington, where one time I attended one seminar, and they were talking about how to apply for successful SBIRs, and it was very influential, where I really learned how to write an SBIR, and probably this is how I got it. And of course, seek advice from experienced individuals. For instance, in our institution, there's a lot of scientists that went down this road before us, and very successfully they have spun out many companies. So we sit down with them, we drink coffee with them, we stumble upon them on the corridor, and we just ask them questions, and they will guide us. So what are we doing currently at Chimerocyte? So as I mentioned earlier, we're evaluating our clinical ability, our highly sensitive chimerism quantification, to be able to identify those high-risk patients that are in risk of leukemia relapse, and correlate that to minimal residual disease, which is a lot of companies right now are doing the same thing. And it's like a highly sought path, this minimal residual disease. So I think that we and others are, at the right time in developing this technology. We're also doing more research and development. So we're creating many more assays, as I mentioned earlier. And after we complete and validate and optimize all our assays, which are not easy to do, they can be done. We have been doing this for years now. After we optimize all those assays, we can go live, and we can have partnerships and go commercial. All right, so what's next? We're exploring 3 commercial options. The first option is to create a commercial diagnostic kit, for chimerism. Our primary customers are going to be transplant center and clinics and research labs. This path will be FDA regulated, so it's going to require a lot of money and a lot of investment. The second path is to create an in-house service in which customers will send us directly their samples, and we can, in a single lab, run them and test them for them. This is going to be not regulated by the FDA, by the CMS, which is the Center for Medicare and Medicaid Services. And the third path is to license our technology to larger clinical centers. And for now, this is a preferred path that we want to take because it's the easiest and the less costly. And also our technology can be easily applicable to clinical centers where they have the right equipment. I mean, they have qPCRs and all the equipment needed to do so. So if I want to give any advice for academics that want to go the route of entrepreneurship, well, they need to be motivated and enthusiastic because it's another beast to be doing science for commercial applications. And you have to have your $1 million idea, and you have to really love it. And this is my case, actually, so I'm pretty happy with that. Of course, seek help and support. This is very important. And to be honest, I was really surprised of how much my institution was willing to help me. And it's like everyone wants you to succeed, and that's very critical. Don't hesitate to seek help at your institution. All right, I'm just gonna thank you all. This is me and my partner at the company. Right now, she's not here with us, but she has been a huge influence on me, and without her, this would not have been possible. All right, so thank you all. I am done with my presentation, and I'll give back to Daniel.
Great, thank you. So for Kory and Sami, please turn on your webcams. We are going to transition to the Q&A phase. So please start submitting your questions in the chat box. We'd love to hear them. So one question that you touched on here, Sami, that I think is a good one for the scientists on our call is, how easy was your transition from academics to owning a small business?
So for me, it wasn't as hard as I thought it would be, just because we had such a great support at our institutions. So again, as I said, don't hesitate to ask. There are, in your institution, there's dedicated departments that help scientists to transition their technology to a clinical application or to business, and go and ask them, and they will help you.
Great. Kory, I think this is a question for you. Is the AAP open to contract applicants? Oh, Kory, you need to turn on you're muted right now. Sorry.
Sorry, thank you. We're always trying not to be disruptive to other presenters, right? Yeah, so not right now. The AAP is only open to grant applicants. Contractors would have to submit it themselves.
Great. So let's see. I have a couple other pre-submitted questions. Do you have any specific advice on how-to statements on gaining the initial startup funding for launching a new business?
Sorry, I was reading the chat. Can you say that again? There's an LLC question in the chat.
We'll get to the LLC question next. So do you have any specific advice or how-to statements on gaining the initial startup funding for launching a new business?
Oh, well, so I can talk about SBIR and STTR. Sami, I don't know, did you have funding pre-SBIR?
No, actually, the SBIR is has enough margin for our company to be able to cover all costs that we're currently doing. So currently we don't need any more funding for now.
So we'll get to the next question, which is an LLC company qualified for an SBIR grant?
Yes, we frequently see people with LLCs apply for SBIR. So the eligibility criteria are that the company is organized for profit, that it's a small business by federal standards, and then also that it's US owned and operated.
Okay, so I think this is a follow-up to the AAP question. Have the contract opportunity topics been finalized yet, and when are they usually finalized?
So the R&D contract opportunities, those are specific to SBIR. STTR companies or projects are not eligible for R&D contracts. They usually come out in late July or early August. You won't hear us talk about them because we're not really allowed to, unlike grants that we're allowed to talk about them ahead of time. Contract opportunities, we have very limited communication that we're allowed to give applicants. And so look for that in July or August. Once it comes out, the NCI SBIR program will be announcing the solicitation. We should have links everywhere in e-blasts and everything else.
So here is another question. How advanced should your proof of concept be for your research to be considered for an SBIR or STTR?
It's a good question. I would encourage you to call a program director and talk to them because it probably depends on technology area, how crowded the field is, and a number of other things. So I would encourage you to put together something, either an abstract, executive summary, ideally a specific aims page, and send it either to me, or you can look on our website. All the program directors are listed with their contact information and their portfolio areas. So send it to someone and ask to schedule a phone call.
Okay, so how old does the company have to be for a grant application, and approximately how many are funded each year?
It just needs to be a company. That's it. I think, Sami, if I'm not mistaken, you were pretty early when you applied for an STTR, correct?
Yes, I was writing my grant and creating the company from scratch at the same time.
Yeah, so it has to be a registered company.
Yes.
And just note that to apply, there's a series of registrations that you have to complete. I think there's 5. They take a long time, and they kind of have to be done in a sequential order. So we encourage people to start on their registrations up to 8 weeks ahead of the deadline.
Yes.
Was there another
And
part to that question?
How many are funded every year?
Oh, how many are funded every year? We have 400 active projects at any time. We get about 1,200 to 14 applications a year, and we fund somewhere around 10-ish%, so 140, 150 applications.
So here is a question for Sami. Are you still working as a scientist at the Institute, or are you full-time for running the small business?
Oh, absolutely. I'm still working at the institution. So I love working science a lot. And I also love this entrepreneurial aspect. So, and this is the beauty of the thing. We can still do both of them at the same time. So the institution here has some sort of a policy that when the company becomes big enough or have a lot of enough money, then they will make you choose either stay with us in the institution or go full-time company. But at this stage, they will support us, and we can still be at the company and at the institution. And I also work on many projects, pure science projects with Lee.
So we have a couple of questions about ownership and citizenship and localization for the parties involved. So we have one person asking whether the owner of a company should be an American citizen. And then we have a very interesting project that looks like it is being produced in both Argentina and Nigeria. So I guess what are the citizenship and localization requirements for applying for SBIR or STTR awards?
Yeah, so there's a couple of things here. So one is ownership. Another one looks like where the work is being done. So ownership, the ownership of the company, it must be owned, the majority of the ownership must be from U.S. citizens or permanent residents. And so if you are not a U.S. citizen or permanent resident and you own 51% or more of the company, then you would not be eligible for SBIR and STTR. You may still be eligible for other types of grant funding like R21s, R01s. Oftentimes small businesses can apply to those. So that's something to look and always read the funding announcement and look for the eligibility criteria there. Now, as far as work being done outside of the United States, again, this is very difficult. This is not as strict of a rule as the ownership, but it for almost everything, the work and all the supplies and all the work must be completed in the United States that is funded by the SBIR. So all of the grant money needs to be used in the United States unless there is exceptional scientific justification. Usually the exceptional scientific justification is pretty difficult. It's usually easier to work around than it is to get the approval. With the exception, we have a funding announcement right now for technologies to help cancer patients in developing countries and also in rural parts of the United States. And so in that case, it may be easier to do work outside the United States, especially with patient populations. So if you have sort of a global health technology and a small business, look at our website for that funding announcement.
So this next question, I guess would be for both of you, because Sami, I don't know where exactly you are in your funding, but could you share some of the critical points during the bridge phase of the transition, I guess, to commercialization?
Yeah, I can. Sami's not quite there yet, but I guess I could have let you say that. So for the bridge, we often see people doing IND enabling studies. I'm less familiar with the devices milestones for development, but I think probably at the IDE phase, don't quote me on that, certainly. But for therapeutics, often IND enabling, sometimes people are doing some early clinical studies or they're doing part of a clinical study. So if you're seeking bridge funding and there's going to be a co-funding situation where the business is going to be paying for part of the project and the grant's going to be paying for part of the project, again, I suggest you call the program director when you're developing your application to talk about how you should represent that in the application, because we have knowledge of what works and what doesn't historically.
So here is a question about funding pay lines. I guess if you have the choice between an SBIR or an Sttr, which one would you go for? So it looks like the SBIR has more options in the Sttr or higher pay line than the Sttr budget, but are they very different?
So as I mentioned in my slides, usually it is the PI's employment status and then how much work will be done at, if you have a not-for-profit or a university partner, how much work needs to be done at that site. If you can build the project to go either SBIR or Sttr, because some people can do that, I would call, you're going to keep hearing this from me, call a program director. And the reason I say that is the Sttr budget, the Sttr set aside is much smaller. And so we see much more fluctuation in the Sttr success rates from year to year. And so for this year, for example, and I think last year the Sttr budget was much tighter than the SBIR budget. And so I would probably encourage you to go SBIR if you can. But you should never try to make a project fit a path that doesn't really work, because the peer reviewers will notice that and it'll affect your score.
So here was a question. How do you, and this would be for Sami, how did you convince your university to support your business idea?
It wasn't hard at all. We just had the idea. We talked with the business development strategy department, and they just literally took me by the hand and showed me all the things that I need to do. And yeah, so as I mentioned, the supportive environment of your institution is very important. So it could depend on your location. So Seattle is really supportive. There's also other cities. Boston is one of the best, I hear, where there's a lot of biotech companies that come there. So it can depend on the location, but here in Seattle, it's very, very supportive.
We did have someone submit a question also. Do you have any advice in finding collaborators for your small business grants?
So my collaborator was located 2 floors above my lab. So I just walked to him and talked to him, and he was like thrilled to collaborate with me and provide me with patient samples to test. So this is how it happens. Also depends on the institution.
So we have someone who asked a question. I'm an academic working with a small company that has both for-profit and non-profit aspects. Which would be the best to fund our R&D, SBIR or Sttr?
So both the eligibility for both the SBIR and Sttr programs, the business must be for-profit. Really, deciding between SBIR and Sttr boils down to if you have an academic research partner, how much work you want done at an academic site. And that may be what you mean on the nonprofit. So I guess I'm not clear if you have like a for-profit and a nonprofit arm. But if the not-for-profit needs to do a lot of the work, then Sttr is the right mechanism.
So here's another question. This person who wrote in was interested in if there was any way to ask for SBIR funding when all the research is being done on a university campus or in a university campus lab.
So no, there are minimums, and under the SBIR program, 2/3 of the work needs to be done for a phase 1 at the small business, and under the Sttr program, 40% of the work needs to be done by the small business. So if 100% of the work is going to be done by the academic institution, then R21 or R01 funding is a better.
Okay. Well, let's see. Well, I'm going to see if there's any more questions that are going to come in. But Kory, Sami, are there any last words that you would like to share with our attendees?
Any last words? I would tell them that if they have this idea, they don't need to hesitate. Go for it. I have had an excellent experience, and yes, so go for it if you have it.
So I'll say 2 things. I'll say no, NCI doesn't publish our pay lines. That was a question that just came in the chat. You can call us, and once you have your score, contact your program officer, which will lead me to my parting words, which is don't hesitate to contact us. People, especially people that are new to applying for federal funding, maybe are a little apprehensive about contacting program officers, but just contact us.
Great. Well, thank you all so much for joining us. And I'd like to thank Kory, Sami, and Jenna for your time, as well as Lisa Yom, the communications specialist at the NCI SBIR Development Center, who helped us put together this amazing presentation. I'd like to thank you all for joining us today. The ASBMB provides professional development programs based on your needs, and we hope you'll take the time to fill out a short survey about this webinar. The survey will be available upon meeting exit. Have a great day, everyone. Thank you.